Hello, everyone. My name is Rupak Farahani, Marketing Manager at Advanced Cell Diagnostics, and your host for today. Welcome to ACD's Knowledge Resources webinar series, where we'll give you closer understanding of how RNASCOPE technology is contributing to specific application areas. We begin today's knowledge series on the topic of inflammation, presented by Dr. Anushka Dikshit. Dr. Anushka Dikshit is an application scientist with Advanced Cell Diagnostics with background in cancer research and immuno-oncology. She is focused on expanding the applications of RNASCOPE technology on oncology research. Prior to working for ACD, Anushka was involved in identifying novel therapeutic targets for melanoma as a postdoctoral fellow at Duke University. She obtained her doctoral degree from Southern Illinois University School of Medicine in molecular physiology. With that, we will get started.
So thank you for joining the webinar today. As Rhonda and Pupak mentioned, today we'll be learning about the role of RNASCOPE in expanding applications in inflammation research. So to start off, I want to introduce you to the RNASCOPE technology and go over the basics of using this technology for your research. Next. So RNASCOPE technology is an in situ hybridization assay that was specifically designed to overcome some of the traditional limitations of the ISH assay while still maintaining high degree of sensitivity and specificity of detection. Next slide. This assay was specifically designed to visualize target genes and biomarkers with a spatial and morphological context. The RNASCOPE ISH technology is unique mainly because of its target probe design strategy, signal amplification system, and detection and quantification system. ACD will design a specific target probe against the region of your interest or your target gene of interest. We have a signal amplification strategy which allows you to visualize your target with high degree of signal to noise ratio. And our detection and quantification system allows you to visualize a single molecule of RNA as a punctate top, either with chromogenic or fluorescent signals. In addition, our technology allows you to visualize targets like mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, short targets, point mutations, now microRNAs, siRNAs, ASOs, and so on. Additionally, with our technology, you can visualize targets in SSPE tissues, fixed and fresh frozen tissues, PBMCs, and cultured cells. So because we allow high degree of flexibility in target detection and sample type detection, our assay has been applied to a wide range of applications. Next slide. In this slide, I want to go over the probe design strategy and the signal amplification system that makes this assay unique and what makes it stand apart from some of the other tools for gene expression analysis. So to start with, as you can see on your screen, we have the RNASCOPE probe depicted as a Z molecule. The target binding region is where your probe will bind on to your target transcript of interest. You have a pre-amplifier binding site, which is where you will have the amplification tree forming. And then you have your linker region that connects these two regions of the probe. As depicted on the screen, we design our probes as pooled probes of approximately 20 ZZ pairs, by which I mean 2 Z probes have to bind in tandem onto your target transcript of interest for the signal amplification tree to form. Next. As you can see, the Z probes form a foundation for binding the pre-amplifier onto the double Z pair, after which, next, we have the amplifier molecules bind on to the preamplifier. And next, you will see the chromogenic or fluorescently labeled probes binding onto this amplifier, giving you this amplification or signal tree. Now, this allows for very high signal to noise ratio, and it provides detection of even the really low expressing target transcripts. Next, how do we ensure specificity of target detection? As you can see on your screen, we have depicted that when 2Z probes do not bind in tandem to your target transcript, then the preamplifier is not able to bind onto the Z pair, as a result of which you do not have your labeled probes binding onto the preamplifier, and there is no signal detected. So this ensures high degree of specificity, and our amplification strategy ensures high degree of sensitivity, giving you high signal to noise ratio. Next slide. So in this particular slide, we have depicted all the three assays that fall under the RNASCOPE technology umbrella. We have our RNASCOPE assays, our BaseScope assays, and our microRNAscope assays. The RNASCOPE assay is specifically designed for targets that are over 300 nucleotides in length and are normally either in chromogenic or fluorescent platforms. We have flexibility in plexing for up to 12 plex, where you can visualize 12 targets simultaneously. Then our second assay is our BaseScope assay, which is our specialized assay to detect really short sequences that are up to between 50 to 300 nucleotides in length. And these are mainly to detect your splice variants, highly homologous sequences, and point mutations. And this is available as a single plex and duplex assay, both in automated and manual formats. And our last assay is our microRNAscope assay, which is again a specialized assay to detect really short targets. And as the name suggests, it's used for detection of ASO, microRNAs, and siRNAs. Currently, it is available as a single plex in automated and manual formats. In this slide, we have shown you the breadth of our product portfolio, wherein we allow flexibility in plexing with RNASCOPE, where you can detect single, duplex, and up to 12 plex of targets simultaneously. Then we have our BaseScope assays, where we can detect 1 to 2 targets, and our microRNAscope assays, which can detect a single target. Currently, we have over 2,600 probes in our catalog, and if we do not have the target of your interest, we can design a probe against any target of any species within less than 2 weeks. Now let's focus on today's topic of interest, which is to look at applications of RNASCOPE technology in inflammation research. Tissue inflammation normally encompasses increased influx of immune cells in target tissue. Now this can happen in injury, in diseases like cancer or autoimmune disorders, and so on. With our technology, we can visualize target immune cells and the secreted chemokines and cytokines in your target tissue. Identifying pro-inflammatory cytokines can help us determine the immune cellular source of those soluble factors and potentially can help us develop therapeutic strategies for autoimmune disorders, infections, and cancer. In today's presentation, I'm going to show you various examples in different research areas where RNASCOPE technology has been used to visualize target immune populations and soluble factors like chemokines and cytokines that are causative agents for a lot of these disease and injury models. In this first example, we have looked at the expression of immune checkpoint markers like PD-1, PD-L1, LAG-3, TIM-3, CTLA-4, and PD-L2. We have compared the expression of these immune checkpoint markers in 2 lung cancer tumors from a tumor TMA. Here we are looking at core 2E2, which has high degree of expression of these inflammatory immune checkpoint markers and inflammatory cytokines. And then we have core 2F1, which has low degree of expression of these immune checkpoint markers. This is again shown in the quantification data in the bottom of the page. What we wanted to see was how was the degree of expression of these immune checkpoint markers affecting the immune cell infiltration in these lung cancer tumors. So here we've compared the expression of certain immune cell markers between these two tumors. And as you can see, the 2E2 core shows high degree of expression of markers like interferon gamma, CD8, FOXP3, CD4, CXCL10, CD68, and so forth. Now, the markers mentioned here are specific for cell types such as cytotoxic T lymphocytes or T regulatory cells or M1 and M2 macrophages. What is shown here is that the core 2E2, which showed high degree of checkpoint marker expression, is also showing high degree of immune cell infiltration compared to the 2F1 core. This indicates that this strategy can be applied for patient samples wherein you can visualize immune checkpoint markers and immune cell infiltration to determine the success of immune therapies like anti-PD-1 or anti-PD-L1 therapy. So by studying the inflammatory signature in these tumors, you can identify the potential impact of these immunotherapies on patient samples. Next, we have an example where these researchers were studying the role of epithelial sodium channels in the respiratory tract. Now, the epithelial sodium channel is expressed in different cells of the respiratory tract, and they were studying the role of the alpha, beta, and gamma subunits that make up this channel, since this is implicated in a number of lung disorders that are triggered by lung inflammation. So as you can see in this image, by using the RNASCOPE duplex assay, they were able to use probes for the alpha, beta, and gamma subunits of this sodium channel and visualize the expression of these markers in situ. Using a overexpression model, they were able to show that when the alpha subunit was overexpressed, they could see a visible change in the pathology of the parenchymal layer of this lung tissue, whereas if gamma subunit was overexpressed, they were not able to see this change in the tissue pathology, thereby helping them understand the role of this alpha subunit predominantly in causing changes in the pathology of this lung tumor, which leads to increased inflammation. In this next example, these researchers were studying the effect of flu infection on tissues other than the lung. So it is the effect of infection of the flu virus on the lung tissue is very well studied, but the manifestations of this disease on tissues other than the lung are poorly understood. So in this example, they used the ferret model to study the impact of the avian influenza A virus on lung pathology. So they have used a singleplex assay and a ferret model to show that when they inoculated this avian virus with the ferret liver tissue, they were able to see that this liver tissue showed increased expression of IL-8 both by the fibroblasts and the endothelial cells. So this gives us an idea about the role of or the effect of influenza infection on extrapulmonary tissues and help them narrow down on specific cell types that were producing these soluble cytokines. In this next example, they wanted to study the role of secretory cytokines like IL-6 and mainly TNF-alpha on ovarian cancer carcinogenesis. So here they're studying the expression of TNF-alpha in the clear cell ovarian carcinoma tissue to show that specific cell types are expressing or releasing this TNF alpha and potentially contributing to the carcinogenesis of this ovarian cancer sample. So using both our duplex and singleplex assays, they were able to show that IL-16 TNF alpha were sometimes produced by the same set of cells, but were also independently produced by different cells of the lung cancer tumor. The ovarian cancer tumor. So here is a very exciting paper that came out last year where they were studying the role of gamma delta T cells in cancer progression. So gamma delta T cells are known to be one of the first population of T cells to mount an anti-tumor immune response. But they've seen that as the patient ages, there is a change in the phenotype of these gamma delta T cells, and they no longer can mount an anti-tumor immune response successfully. So they wanted to visualize and study these gamma delta T cell populations, for which they used a mouse model and studied the expression of these cells in young and old mice. What they were able to show was that as the mice age, the gamma delta T cell population changes to produce increased level of IL-17 cytokine. So these IL-17 producing gamma delta T cells are a more suppressive type of T cells that are not able to mount a strong anti-tumor immune response. As you can see in this graph, the levels of gamma delta T cells in the older mice are higher than in the younger mice. Using the RNASCOPE multiplex fluorescent assay, they were able to study these cell populations and also identify a set of stromal cell populations that produced IL-7, thereby promoting the growth of IL-17 producing gamma delta T cells. So here they have looked at the expression of gamma delta T cells in younger and older mice, and you can see an increased expression of IL-17 producing gamma delta T cells in the older population compared to the younger population. So here they were able to successfully show the expression of soluble cytokines such as IL-17 and IL-7 using the multiplex assay. In this next example, from a paper published in Nature Communications, they were studying the expression of the fibronectin type 3 domain protein gene expression. This particular FNDC4 is known to be expressed in inflamed colon, especially in patients with inflammatory bowel disease. Now, the FNDC4 plays a role in reducing the activation of macrophages and therefore has a more anti-inflammatory response for infiltrating macrophages, thereby controlling the inflammation in the IBS patients. So using the RNASCOPE technology, they were able to show that in a compared to control sample, animal model with an IBS phenotype shows increased expression of this particular target gene. Next, we have a very interesting paper which shows the role of increased level of cytokines and its impact on macrophage polarization. This is a model to study pancreatic cancer, where using RNASCOPE, they were able to identify that certain cells in a pancreatic tissue that is undergoing transformation from normal to cancerous shows presence of IL-13 producing cells. These include intraepithelial cells and tuft cells. Using the ISH-ISC technique here, they were able to identify IL-13 producing cells and use the IHC marker to mark those tuft cells in this case. So these IL-13 producing cells cause polarization of macrophages, and these polarized macrophages in turn produce IL-1RA and CCL2 to promote pancreatic cancer growth and fibrosis. So we have shown that using RNASCOPE technology alone, you're specifically able to see your target gene expression and assess your gene expression with a spatial morphology and visualize your target gene and cell population within your tissue of interest. While that gives you enormous amount of information, to thoroughly characterize complex and heterogeneous tissues, it is important to have a more multi-omic approach. So with that in mind, our technology allows for combining our ISH assay with ISC or IF, which we call ISH-IF or ISH-ISC assays. What this allows is to visualize your RNA target in combination with your protein target. So this way you can visualize your, you can perform your ISH and ISC simultaneously on the same sample. This technology has been used widely with our RNASCOPE assay in a number of research applications like oncology, neuroscience, cell and gene therapy, developmental biology, and so forth. But today, we're specifically going to look at examples wherein researchers have used this ISH-ISC technique to study inflammatory response. Now, this is a seminal paper that came out in Nature Medicine last year, where they were studying the effect of an anti-PD-1-induced encephalitis in a patient sample. So a patient that was being treated with PD-1 therapy showed adverse events to this therapy and had encephalitis, which was ultimately fatal. So these researchers were really interested in understanding what population of immune cell induced this encephalitis in these patients. So as a result, they performed PCR sequencing in the inflamed tissue to identify the specific sequence that was predominantly expressed in this particular inflamed tissue. Now, they identified the sequence and using our BASESCOPE assay, designed a probe against it. And then using an ISH-ISC technique, they were able to identify the specific cell population that this TCR was expressed on. And this was a CD4 positive T cell population. And they were able to identify this by using an antibody against CD4 and using our BASESCOPE TCR probe. As you can see, these co-localize on the CD4 positive cells and not on the CD8 positive cells, helping them understand that it was a CD4 positive memory T cell population that was inducing this encephalitis. Next, we have an example of how this ISH-IS technology was used to study inflammation in the brain. Rift Valley virus can cause serious infections in livestock, but they are shown to have zoonotic transmissions in humans as well. And humans who get infected with this Rift Valley virus show high degree of encephalitis. So in this example, they were using a rat model to study the encephalitis induced by Rift Valley virus. They used RNASCOPE probes to study the infection by this Rift Valley virus by designing a probe specifically against the viral RNA. So they can visualize the viral infection simultaneously with the inflammatory response mounted by this infection. So they used antibody against IBA1 to study increased infiltration of microglia in these infected tissues. And as you can see, compared to uninfected or post-infection day 1 to post-infection day 7, you see increased level of these viral mRNA expression along with increased levels of microglia infiltration, showing increased post-day infection, you see a higher inflammatory response mounted. In this next example, these researchers wanted to build a cell atlas to compare healthy colon versus a colon of patients with ulcerative colitis. So to build a cell atlas, they needed patient samples from normal and ulcerative colitis tissue. And this was really important to understand the differential gene expression and the novel cell types in an inflamed colon. So they isolated about 300,000 cells from 18 ulcerative colitis patients and 12 healthy individuals to establish the cell atlas. Once they had these subtypes identified, they used the RNASCOPE assay to validate these subtypes in situ. So they used probes for BEST4 and RSPO3 probes in combination with epithelial and fibroblast markers such as EpCAM and Vimentin. They use antibodies for these cell type markers and then combine them with the BEST4 and RSPO3 probes to visualize specific populations of epithelial and fibroblast cells in these healthy colon tissues, thereby validating their single cell data. Similarly, they were able to visualize and compare a specialized inflammation-associated fibroblast population between healthy and inflamed tissue. As you can see, the inflamed tissue has a high degree of expression of this IL-13RA probe compared to the healthy tissue. So what they were able to observe by the single cell data, they were able to visualize that in situ using this HIF technology. In this next example, again, they were wanting to establish in this example from the same paper, they were wanting to study different subtypes of immune cells and visualize them in situ. So again, they designed probes against soluble factors like IL-17A and use antibodies against CD8 and CD4 and compare the expression of these populations between healthy and inflamed tissue. As you can see from this graph, they were able to show that the expression of these novel immune cells was much higher in the inflamed tissue compared to the healthy tissue. Similarly, again, there was another subtype of immune cells that was specialized and was highly expressed in the inflamed tissue that was positive for TNF-alpha, and they were able to visualize this in situ. As you can see, this is a triple positive cell population, and this is a dual positive with no TNF-alpha expression. So they were able to visualize it, quantify, and then compare to their single-seq RNA data. Another example of validation of single-cell RNA-seq for inflamed tissue is in this particular paper, where they established an atlas for the human retina. And their main goal was to identify cell populations and study basal gene expression for targets that are implicated in age-related macular degeneration. Although we understand the changes in target gene expression for these targets in disease conditions, they wanted to establish the role of these targets and the cell populations expressing them in normal retina. So they performed single-cell RNA sequencing and established cell populations in the normal retina. Here they have used an antibody marker GS to mark the Muller glial cells and used ISH probes for FOS, collagen 4A, and FTL, which are independent markers for independent macroglial cells. So as you can see, these markers are expressed independently as visualized by RNASCOPE, and GS is marking the Muller cells. So after validating and establishing these three distinct subtypes of the macroglial cell populations in the retina, they wanted to study the targets that are implicated in AMD. So these are the set of targets that are implicated in AMD, and they designed probes specifically against those targets and visualized them in combination with the GS antibody. And what they were able to see is that these targets implicated in AMD were co-localizing with GS, showing that the cells that are implicated in AMD are Muller glial cells. So using this cell atlas in combination with the known targets for AMD associated genes, they were able to show that the cell populations that are implicated and dysregulated in AMD are these Muller glial cells. Then we have another example where researchers use the RNASCOPE technology to validate single-cell RNA sequencing to study liver fibrosis. Liver fibrosis is accompanied by increased inflammation of the tissues, followed by excessive collagen production that leads to fibrosis. Now here, after performing single cell sequencing, they were able to identify two distinct stellate cell populations in the liver, the portal vein stellate cell populations and the central vein stellate cell populations. Now, NGFR is a marker for the portal vein stellate cell population, and using HIS, they were able to show that these collagen-producing cells were not co-localizing with the portal cells or portal vein stellate cell population. On the other hand, by using markers for the central vein hepatic stellate cell population, they were able to show that these collagen-producing cells were the same as these ADAMTS positive cells, indicating that the cells producing these collagen and responsible for fibrosis are the central vein hepatic stellate cell populations. So what they were able to see and understand through single cell sequencing, they were able to visualize and confirm using HIF technology. So we really understood the role of the HISC and HIF technique in expanding the understanding of complex and heterogeneous tissue. While a lot of researchers have used this technique successfully, we understood that there are certain subsets of antibodies or targets that cannot be visualized using our current HISC or HIF platform. And that is because certain targets are extremely sensitive to the protease treatment in our ISH workflow. And as a result, we have come up with a set of reagents that have optimized the HISC and HIF technique and will allow you to include a larger subset of antibodies to be used in combination with RNASCOPE ISH. So by performing this co-detection of RNA and protein, we are able to expand the research in different application areas, including understanding or detecting pathogens with their host cell markers, by visualizing target genes producing non-coding RNA, by visualizing entire cell border and its target gene, and correlating RNA and protein expression. So what we have done is introduced a set of reagents that optimize this workflow, and we have tweaked the workflow such that you perform your primary antibody incubation before you perform your RNAscope. This ensures really good antibody signal that is not impacted by the protease treatment in RNAscope. So by introducing these set of reagents and by tweaking the workflow, we have designed an ISH-IHC or ISH-IF assay that gives you optimum RNA and protein signal. This assay can be used with can be used in combination with all of these RNAscope assays, and I'm going to show you a few examples of that. But here is our newest workflow. As I was mentioning, the goal of developing these reagents and workflow was to reduce the optimization required by the researchers in identifying the most suitable antibody to combine with RNAscope. By introducing these reagents and workflow, we want to ensure that there is minimum optimization and most antibodies will be compatible with this workflow. So again, you permeabilize your sample, you bind your antibody, followed by which you hybridize with your ISH probes, amplify your ISH probes, then add your secondary and develop both your RNA and protein signals. And we have tested this in-house, and we are confident that researchers will now able to use their favorite antibody without trying to hunt for a suitable antibody that will work with this workflow. So the three reagents that we've introduced with this co-detection assays are the new target retriever, the antibody diluent, and the co-detection blocker. Depending on which RNAscope assays you are interested in, you will need to use one or all of these reagents. The co-detection target retriever allows you to not compromise on your ISH RNAscope signal. The primary antibody diluent is essential for optimum antibody signal. And your co-detection blocker ensures there is no cross-link between the ISH and IHC chemistries. So these reagents have really optimized this protocol, wherein you can perform your RNAscope in combination with your ISH or IS and get optimal RNA and protein detection. Now, here is an example of how we've compared a few previously incompatible antibodies with IHC alone, with our sequential workflow, which included performing ISH followed by IHC, and then with our new workflow with our new co-detection reagents in the third column. As you can see, CD4, CD5, and FOXP3 signal is comparable to IHC only with our new reagents, whereas these antibodies showed some significant loss of signal or non-specific detection with our previous workflow. In this particular example, again, we have shown that CD3, CD8, Ki67, again, very commonly used antibodies, perform really well with our new set of co-detection reagents, where if you compare to IHC only, the CD8 signal that was completely lost in our ISH-IHC original workflow is now retained without compromising the ISH signal. So here, CD8 is a very significant antibody, especially when you're studying immune cells and inflammatory response. And we are now able to make this antibody compatible with our RNAscope ISH assay. Now, this is an example to show you how this is compatible with our V2 multiplex assay. Here we have used our housekeeping probes polar to ATP, IB, and UBC in combination with Ki67 antibody. And using our new co-detection assays, you can perform multiplex V2 assays as well. Again, just a few examples of previously incompatible antibodies, which are significant targets while studying immuno-oncology or inflammation research. CD11C, CD8, CD19, and CD20 are now compatible antibodies with RNAscope, where you do not see any significant loss of signal and you see very clear detection of your immune cells in combination with your ISH target. So this is just a nice table to kind of elaborate on where you would be using these co-detection reagents. And we have two different part numbers for this that you can use based on which RNAscope assay you want to perform. As I mentioned previously, it's compatible with RNAscope Red assay, BaseScope assays, multiplex V2 assays, both in manual and Leica platforms. So what we have really established is, in addition to inflammation research, ISH-IHC is a very valuable tool to expand the understanding of gene and protein expression and answer significant research questions. So this allows you to incorporate your favorite existing antibodies and your IHC workflow with your RNAscope targets, thereby augmenting your research and understanding of cellular mechanisms. Since we allow for the flexibility to perform this either manually or Leica, you can do this with either of the platforms and any of these assays mentioned here. So by allowing and optimizing this co-detection workflow, we really hope that we can enable expanding research in different application areas. So with that, I want to highlight that our RNAscope technology has been very widely applied over the last few years. Currently, we have almost 3,000 publications, and these are in different research areas. But as you can see, cancer, neuroscience, infectious disease are our popular areas where we see a lot of publications. But inflammation research is also up there, wherein we have a lot of publications in individuals studying inflammatory response using our various RNAscope assays. So in summary, our assays are highly specific, will give you very high signal to noise ratio. We have high degree of sensitivity ensured by our probe design and amplification strategy. We allow for multiplexing, where currently you can visualize 12 targets simultaneously in our fresh and fixed frozen samples. and up to 4 targets simultaneously in our FFPE tissues. We're able to combine IHC to show protein and RNA detection on the same sample, thereby allowing conservation of your precious samples. And finally, we also have our specialized assays like PhD and microRNAscope assays that allow for detection of those unique RNA species like splice variants, short targets, and small RNAs. So with that, I would like to take any questions. Thank you.
Thank you very much, Anushka. This ends our presentation portion of the event. Before we open all questions for Dr. Dikshit, I'd like to let you know that we'll have more knowledge resources webinars in upcoming weeks. Our next topic will be on single cell applications being held on next November 2nd, so next week. And we're looking forward to seeing you there. Meanwhile, I'd like to encourage you to visit our website at actbio.com to learn more about our products and services. Please feel free to contact our sales and support colleagues if you have any questions or if you'd like to discuss your projects. Thank you. Rhonda?